Hello, and this is going to be a quick video on Unit 4B, which is talking about movement across the cell membrane, either passively or actively. First thing is we're going to talk about the concentration gradient. As you can see, we've got a lot more of these greenish, yellowish dots on this side than we do on this side, and normal motion would be for the high concentration of solute to move from where it's high to where it's low. And you can see it on this section too. We've got more of the green blobs, high concentration, and the natural motion is to move this way. How concentrated the molecules are naturally moves from high to low concentration. Okay, the first type of transport we're going to talk about is passive transport. Passive transport can be done a couple of different ways. First way is diffusion, and this is simply moving from high concentration to low. It's movement from high concentration to low. So diffusion is moving any kind of thing from high concentration across this this cell layer, this double lipid layer, from high concentration to low. We can also have what's called facilitated diffusion. If the space is too small for the molecules that are moving through, we can have pore proteins and or channel proteins, where the pore actually allows the larger molecules to move, again from high concentration to low, requiring no energy from the cell whatsoever. Or this channel protein that opens up and lets one in and then lets one out, and then opens up and lets one in, closes top, lets one out through the bottom. So facilitated takes molecules that are too large and moves them from high concentration to low, and again requiring no energy from the cell. Okay, I'm going to show you a short little video talking about this plasma membrane and also moving this pore protein.
Some membrane proteins, called ion channels, provide a pore for a specific type of ion to pass from one side of the cell membrane to the other.
You can see the sodium ions,
this
plus
passive
ions,
transport, which requires no expenditure of energy, is an example of facilitated diffusion. In this example, sodium ions move spontaneously down their concentration gradient from an area of higher concentration to an area of lower concentration. Because ions are electrically charged, their movement is also affected by any difference in charge across the membrane. Here, the inside of the cell is negatively charged relative to the outside. The positively charged ions are attracted to the negatively charged interior. The two factors, a difference in electric charge and a difference in concentration, create an electrochemical gradient down which the ions flow spontaneously.
Okay, examples of passive transport. You can see there's respiration. This is when anytime the gas moves from the alveoli, from where we're breathing the oxygen in and we're exhaling the carbon dioxide. So the capillaries bring in red blood cells, and I really wish they would never show blood cells as being other than red, because they are red. If you've heard red blood is blue inside the body, it's 100% false. So red molecules move in carrying carbon dioxide, and then through diffusion, where the carbon dioxide is higher in the blood than it is in the lung, it moves right into this alveoli, this small little lung bubble that you see right there, and then oxygen diffuses out and moves out to the body so it can actually give oxygen to all the cells. Then it moves back in. You exhale the carbon dioxide, you inhale oxygen for this whole process to work. Also works inside of your stomach. Your small intestine, which is bringing food in, has blood moving in, and it actually picks up food through these villi, and you can see the microvilli on top of the villi. It increases the surface area. Blood vessels come in, and material from the small intestine can actually diffuse in food, carbohydrates, lipids, proteins, whatever we get from our diet, and then it moves out to the cells so it can be utilized. So monomers from food travel into the villi, from the small intestine to the blood vessels, and then off to the cells. Another example is the circulatory system, where we actually get red blood cells moving through carrying oxygen, and the oxygen can go to the cells, as you can see, and we have material going from the cells to the red blood cells so it can get carried to the lung. So assume that Z is the oxygen and the Y is the carbon dioxide, and the X is going to be something else, maybe carbohydrates. moving out to it. And urea would be moving back into the blood so it can go to the kidneys. And another example of passive transport. Kidneys filter out toxins from the blood. It regulates the water balance. And you can see clean blood, actually the blood with the waste products goes in. It comes into and actually gets filtered out into these little tubules inside the kidney, and then goes out as clean blood, so it can actually pick up more waste from all the cells to start the process all over again. And your kidneys, of course, bring that yellow material, the urine, to the bladder, so it can actually be given out past the body, because urine is a poison. Here's an example of osmosis. Got water on both sides. We have a semi-permeable membrane in between. And you can see that the water actually moves from one side to the other. And if we add some salt to this side, now we have less water on this side, and the water moves over here, and what happens is this level drops and that level goes up. That's osmosis, diffusion of water from where it's high to where it's low. Here's another example of osmosis. Selectively permeable membrane on this side. We have less water, more solute, and we have more water, less solute. So what should happen is the water should move in this direction, trying to reach equilibrium. In other words, keep the space between the molecules, the purple molecules on both sides. And you can see it's done in this location where the water moves out, so we can actually raise the water level on one side by adding solute to that side. Then we have what's called active transport. Active transport is in requiring energy from the cell, and this is ATP, the adenosine triphosphate. It can be anytime we bring in large material from the cells, where we actually bring in large material. We throw in digestive enzymes or lysomes to break it up. We pull out what we want, and then we take the other waste food in another process where we move it out of the cell. Endocytosis in and exocytosis out. So endocytosis is bringing food or other large molecules into the cell. Exocytosis is getting rid of waste out of the cell. We also have a protein pump, which looks like this, which looks very much like those channel proteins, except that we now have to add ATP into it, because we're bringing things from a smaller concentration to a higher concentration. And anytime we do that, that's against the natural flow, and we have to add in energy. Here's an example of endocytosis and exocytosis, and I'm going to push play and just let him talk to you.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis.
Notice the cell membrane goes
if the
completely
material
around
the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clavulin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
Okay, so again, endocytosis, we have three different types. Large particles come in through a process called phagocytosis. Cell membrane wraps around it, goes into the cell. Anytime we have smaller molecules, usually a liquid, it's called pinocytosis, and again, goes inside the cell membrane, gets cut off, moves inside the cell. And then receptor-mediated endocytosis, where we have these proteins on the outside that pick up the material and then eventually become more concave and move out, completely surrounded by the cell membrane. Here's some examples of all three of those. This is a amoeba. I'm actually bringing in a large molecule, wrapping around it, phagocytosis. Here is material from the outside of the cell brought in, usually a liquid, pinocytosis. And here are the receptor cells, which actually are bringing in material in a process called receptor-mediated endocytosis. Examples of active transport: immune system. Is not only made up of red blood cells, it's also made up of white blood cells, and these white blood cells can actually move out. If you get cut or injured and some sort of pathogen moves in, your body will make histamines, which will actually attract these white blood cells, and these white blood cells will actually phagocytosis, actually take these molecules, these pathogens, and actually release enzymes inside to break it down into monomers, so your body can actually utilize that bad material as a good thing. So white blood cells destroy invading pathogens. Another example is potassium inside and sodium outside, where we actually need potassium on the inside, but we find out that there's more of it outside than inside, and what happens is that we actually bring in the sodium ions, add a little bit of ATP, they move out, it opens up this side, then we can get potassium into the cell, and we'll actually keep doing that over and over until the cell gets as much potassium as it possibly needs. Here are a couple of YouTube funnies. Last slide, 35 seconds. I guess I'll go ahead and show you. This is going to be a paramecium, so we can actually talk about the paramecium down here. So the first one is a paramecium. And let's go ahead and maximize this one. And I wanted you to see the cilia that's moving through. This is the actual mouth of the paramecium, and it's going to be using the cilia to bring these small little objects in. It brings water in, and there's the stomach. So we'll actually bring that material in. If we could actually see one of these go in, that would be really cool. Most of these are really teeny tiny itsy bitsies, which you can see there, and they're actually moving in relation to the cilia. So they're actually all moving into the mouth, and there's another one where the mouth is on the inside. Okay, and let's get rid of this one. Go back to our PowerPoint. And let's do this one. This is amoeba and paramecium, and this is going to be actually moving them into a process called endocytosis. Come on.
I believe I was born to make coffee.
And I love the way they get these ads in.
I was born to make coffee.
Okay, amoeba eating 2 paramecium. This is an amoeba. It's called amoeboid action, where it actually takes his cytosol and pushes out and moves in a direction. There's a paramecium, and here's another paramecium. Not sure where all the kid noise is. I think this is a video in a museum. Now it's totally locked in. It releases digestive enzymes, and notice the paramecium start to freak out. And these digestive enzymes actually break down the cell wall of the paramecium, turning it into mush. And you can see the 2 mush blobs are ending up being 1 mush blob. And now this amoeba can actually take in the monomers, the carbohydrates, the lipids, the proteins, and the nucleic acids from that material. Okay, that does it for this video. I hope you understand a little bit more about cell membrane and how that works. Talk to you later.